This method can help answer key questions in the field of neuroscience, such as determining the temporal relationship between multiple brain regions or exploring the detailed neural pathway of complex cognitive processes.
The main advantage of this technique is that it allows for the integration of multimodal EEG and functional MRI data in a spatial and temporal specific manner to improve the resolution and accuracy of functional neuroimaging.
The implications of this technique extends toward the understanding of epilepsy and neurocognitive disorders, because the methods improves on the spatial and temporal accuracy for non-invasive neuroimaging.
Though this method can be used to provide insight to the visual motor and emotional processing systems, it can also be applied to other systems, such as sensory motor integration, language, and memory processing. Visual demonstration of this method is critical, as the concurrent EEG and fMRI data recording steps can be difficult to learn, and also because of the requirement for specialized facility, equipment, and signal processing methods.
To begin this procedure, place an appropriately sized passive MRI-compatible EEG cap onto the subject's head. Position the electrodes as per the 1020 International Labeling System. In the EEG recording software, check the impedance of the ground and reference electrodes. To do this, click on the Impedance tab and select the electrode type on the software user interface. Next, use a syringe to inject electrolyte gel into each electrode. Then use a cotton swab to spread the gel to ensure skin electrode contact. Once the EEG cap preparation is done, have the subject move to the MR scanner. Set up